Earth, the third rock from the sun, our blue marvel. Sure, it may not be as sexy as Saturn with its icy rings, or Jupiter with its imposing red spot, in which, by the way, you can easily fit Earth and have some wiggle room left. But Earth is the only planet in our solar system where we know life exists, and not just any life, us. Our planet's pretty big, and over 70% of it is covered by water. Through natural or anthropogenic forcings, our planet is constantly changing, and the ecosystems we depend on are too. And understanding how our ocean varies is really important, as water is essential for our survival, and we depend on our ocean for food, recreation, and economic development. This animation is derived from the Eco-Darwin ocean biogeochemistry model and shows surface winds and carbon dioxide flux. Blue areas is where CO2 is being absorbed by the ocean, and red areas is where CO2 is coming out of the ocean or is being outgassed. Those gray lines, or the path lines, indicate surface winds, and winds are one of the drivers of this air-sea CO2 exchange. Now, our planet is pretty big, which is why it's important to observe it from space. And regularly observing our ocean allows us to detect changes that occur over time, which we may not notice if our observations were sporadic. The ocean, much like our weather, changes on a wide variety of timescales, from daily to subseasonal to decadal. And only by regularly observing the ocean can we start seeing a pattern emerge, which is this red line. The pattern, or long-term trend, is induced by climate because the changes are induced by climate. Historically, our climate has varied several times in response to global forcings. These forcings, for example, can be by changes in the Earth's orbit and rotation, or the increase of greenhouse gases in the atmosphere. This animation on the global distribution of aerosols shows how they're carried out across the globe by large-scale weather patterns, and they can produce global impacts. Aerosol species include sea salt that made it over the oceans, dust from desert regions, smoke from fires across the globe. And we can see the 2019 Australian brush fires in this animation, which had devastating consequences on those ecosystems, and whose impacts really went far beyond the area where they happened. Now, we've said that detecting how anthropogenic and natural climate changes influence the biosphere is impossible without time-resolving measurements collected over long periods of time. Satellites are really our unique tool, which provide us with a way to monitor our planet synoptically at high temporal resolution. This is why NASA continually invests in missions to keep our planet under observations to ensure that there are no data gaps. NASA's Earth Science program uses 17 cutting-edge missions in orbit, which include instruments mounted on the International Space Station. And they collect and analyze data that informs the scientific community about our global environment and ecosystem and how they are changing through time. 20 years of looking at our global biosphere has allowed us to see how life on the surface of our planet has changed. We can measure how plants grow on land and in the ocean, and how they help sequester carbon from the atmosphere. This data visualization represents 20 years worth of data taken primarily with CWIS modems and the VIR sensors. In the ocean, the dark blue or violet areas represent areas where there really is not much going on. There might be not phytoplankton or anything else because of lack of nutrients. But the greens and yellows and reds represent areas where there is stuff, including phytoplankton and dissolved and particulate organic and inorganic matter. These areas include coastal regions where cold, nutrient-rich water rises from the depth, and they stimulate primary production, or areas at the river mouths like the Congo and the Amazon, where these rivers discharge nutrients, dissolved and particulate organic matter into the ocean. On land, the green represent plants, such as forests and grasslands, while the tan represents deserted areas like the deserts of the Middle East and Africa, and the whites represent the snow cover and ice in the poles. But remote sensing is not the only tool that NASA relies on to understand our changing ocean. Remote sensing provides us with a remote measurement of what is happening on the surface of the planet from a vantage point that would be impossible otherwise. But in situ measurements are fundamental to validate our satellite remote sensing measurements and complement such data with other biological and biogeochemical measurements that we cannot still do from remote sensing. The Ocean Biology and Biogeochemistry program within NASA's Earth Science Division has been supporting for years the remote sensing and in situ measurements that help us achieve a better understanding of our ocean biology, biogeochemistry, how ecosystems have changed, and what may be in store for our oceans in the future. With better information, we can make better decisions on how to manage our ocean and protect the health of our ecosystems. One of the areas of investment of the Ocean Biology and Biogeochemistry program is in the detection of marine debris from remote sensing. This is a much needed effort, but it's still in its infancy. We really don't know what can be accomplished. 3 projects have been selected recently to carry out activities that are going to focus on understanding what can we assess from remote sensing in terms of marine debris. So they will focus on the assessment of the effects of marine debris on ocean color signals and the remote sensing detection and identification of plastics in the aquatic environment. We know detection of dry plastics is feasible, but in the aquatic environment it's a little bit more difficult and complex. Some of the questions that these researchers seek to tackle include: what are the limitations of detectability in current and planned ocean color sensors? Are the sensor sensitivity enough to detect small targets? Do we have the required bands to perform this spectral discrimination? And can light polarization help detectability? And this in particular is thinking about our upcoming missions such as space. Another area of focus where we have made recent investments is in the Arctic. The Arctic has been changing dramatically, and we have recently selected through our interdisciplinary research and ocean biology and biogeochemistry competition research projects to further our understanding on how these changes are impacting Arctic ocean biology and how we can better characterize the bio-optical properties of this region to improve satellite remote sensing retrievals of Arctic ocean biology and biogeochemistry, and obviously their changes. The research follows on years of study of Arctic change. Research that has been ongoing is from Sam Laney from Utsjoel Oceanographic Institution and his team. They have recently completed a 3-year study into riverine contributions of biogeochemically relevant terrestrial material into the coastal Arctic Ocean, with the scope to understanding how optical approaches might be better used to discriminate and quantify the timing and magnitude of both dissolved and particulate matter that comes from land. They utilize landers and ice buoys with optical sensor packages in the sea ice to measure the river-transported material from the land to the coastal ocean. The landers and the ice buoys also provide valuable information on the longevity and robustness of these commercially available platforms in these cold polar waters. They are continuing this work to develop a better understanding on how terrestrial and freshwater plumes associated with spring fresheet affect sea ice physics, biology, and biogeochemistry. This information is useful in advancing NASA's Arctic colors. The study is still at its scoping stage, and it seeks to quantify the coupled biogeochemical and ecological response of the Arctic nearshore system to rapidly changing terrestrial fluxes and ice conditions in the context of short-term environmental change and long-term climatic change. We're really hopeful that we'll be able to get Arctic colors off the ground and into the Arctic in the future. We're also looking to advance the measurements we make with new sensors and platforms. One such investment is the GLIMMER, the Geostationary Littoral Imaging and Monitoring Radiometer. GLIMMER science and applications emerged from GeoCape, which was included into the 2007 Earth Science Decadal Survey and focused on air quality and ocean color. GLIMMER is an Earth Venture instrument, part of NASA's program that enables PIs to propose satellite instruments to accomplish a scientific investigation. GLIMMER targets the Gulf of Mexico and other coastal waters of North and South America. It was selected in 2019, and work on GLIMMER is just getting started. GLIMMER will be in a geostationary orbit, which will enable temporal resolution observations, roughly hourly, and with a spatial resolution of 300 meters at nadir. It will have hyperspectral capabilities, which is exciting as GLIMMER seeks to enable large-scale quantification of rapid changes in phytoplankton growth rate, community composition, and fluxes from land to sea. GLIMMER is anticipated to advance scientific understanding and management of coastal ecosystems that will benefit society. One of the new directions that the ocean biology and biogeochemistry is exploring is using LiDAR to achieve a deeper understanding on the types of particles that are in the ocean and their behavior. LiDAR is a remote sensing method that uses light in the form of a pulse laser and measures the signal that is backscattered or returned from the Earth or the ocean. Utilizing laboratory measurements and satellite-derived estimates to extract from CALIOP data, which was designed initially to measure cloud and aerosol properties in the atmosphere, one of our NASA investigators, Mike Berenfeld, developed the first satellite-observed global dataset on the ocean diel vertical migration. In this figure, areas colored in red or orange have the greatest difference in light scattering, a signal that indicates that significant numbers of migrating marine critters were in those areas. CALIOP data revealed an increase in migrating animal biomass in the subtropical waters of the North and South Pacific and the South Indian Ocean. In the tropical regions in the North Atlantic, biomass decreased. In all but the tropical Atlantic regions, these changes were correlated with changes in phytoplankton abundance. Now, modeling is another fundamental part of NASA investments. Sometimes models are the only tool we have to explore why things act a certain way in the ocean. What drives, for example, species distribution, and they can help us understand what would happen if some things changed. These figures are from two recent papers by Stephanie Dukevich, where she explored through models dimensions of phytoplankton diversity and climate change. In the figure to the left, she explored what drives biodiversity as means of normalized richness in the surface ocean. These results are useful as they help better understand the mechanistic controls of diversity and understand how diversity might change or be altered in the future. On the right, we have what we call time of emergence of chlorophyll. And because of natural variability, the time of emergence of a pattern is not always straightforward. However, Stephanie argues that utilizing remote sensor reflectance at 490 will provide a more sensitive indicator of change. We depend on our ocean. Sustained high-quality observations are really critical to understand how marine ecosystem responds to change and how these affect society. Our Earth is an integrated system, and a forcing on one side will result in change somewhere else. While we've learned a great deal from studying different components of Earth's system individually, we can really achieve a better understanding of our Earth system by studying them all together and how they may change in the future.